Session 1 deals with Beyond Big Data, the originator of sequencing and the limitations and methodological crimes of big data-driven science. As first speaker is George Brownlee, formerly Professor of Pathology at the Sir William Dunn School of Pathology, whose work on sequences of nucleic acids and their biological implications is well known. He is currently a fellow at Lincoln College, Oxford. His PhD supervisor was Fred Sanger, and it's entirely appropriate that he should talk on Fred Sanger, 1918 to 2013, the biologist who worked out how to sequence proteins, RNA and DNA. Thank you, Phil.
Well, good morning, everyone. It's a real privilege to be asked by Uti to... Can you all hear me? You can. It's a real privilege to be asked to talk about Fred Sanger, who I was lucky enough to work with. He mentored me for my PhD some years ago now. And he also stimulated me to write his biography indirectly. So I've never talked to a group of scientists or humanities, people interested in the philosophy of science, and so I'm really out of my comfort zone. Coming to a group who are interested in philosophy, and knowing that Fred Sanger never ever talked about his philosophy to me, and as far as I know to anyone else, I'm really faced with a problem. So what I'm going to do is to tell you about his work, which you may well know about, but particularly about him as a person. And you are going to tell me what his philosophy was. So let me start with my... I press number 1, do I? So my first slide is my title. And as Uti has described, I'm retired from the Dunn School of Pathology, but I'm actually a biochemist by training. And you will know that Sanger, as Uti said, has been awarded 2 Nobel Prizes in chemistry for his work on insulin as quite a young man in 1958, and later on with Gilbert and Berg for work on DNA sequencing. So what I'm going to try and do to you is to tell you something about his... As I said, I have to ask you to invent what philosophy drove him. So the plan of my talk is to describe his work, highlight what was novel, and talk about his ethics, which he definitely had ethical values. And my plan is to talk about his early years, there's a pointer here, his early years, the insulin period, a long time ago now, his RNA period and his DNA period. I only actually knew him in his RNA period, where I was his student and the first of his students to work on nucleic acids. And I'll try and end up with something about the impact of his work, if you like, his philosophy for this audience. So Sanger had a privileged upbringing. He was brought up in a remote part of England, really, in the West Country, and this is the house he was brought up in. So he was his parents had no money worries. His father was a GP. That means he's in general practice. It's the word we use in Britain, GP. And so he had a, if you like, a medical father and a rich mother who had inherited money from her parents, who were rich Lancashire cotton millers. So there were absolutely no money worries in the young Sanger's upbringing. Surprisingly, I only learnt this when I interviewed Sanger many years after I'd worked with him in 1992. Sanger had a strong religious upbringing. He was brought up as a strict Quaker by his father. And these Quaker ethics certainly influenced Sanger's ethics. Although Sanger himself never became a Quaker, he couldn't believe in the fundamental... in the statements in the Bible. He wanted evidence. Sanger always wanted evidence. So After his schooling in initially a Quaker school and then in a private school in Dorset, it was natural his father would send him to Cambridge, where he indeed had been a student, and indeed where both Sanger's uncles had been a student. But Sanger revolted at quite a young age. He was thinking about his career at quite a young age. At the age of 16, he commented later, his father was always rushing around attending to patients and could not concentrate on anything. So he said, this is a later statement, of course, of Sanger, he preferred the idea of having a single problem he could really get his teeth into. And so this caused complications when he went up to Cambridge, because his father had to write another series of letters. That's how you got into Cambridge in those days, around 1940, to move his son from the medical studies to more natural science studies. So, sorry, I'm getting confused. Moving on, right. So, how did his parents react to this minor rebellion of their son? But his mother wrote in an unpublished diary, "We're delighted that Fred has got 7 credits in school certificate." That was the name of the examination they took at school at that time. So he is right for Cambridge. He has decided he doesn't want to be a medic, which is rather disappointing, but we feel it's no use to press him if he doesn't like the idea. So this is an unpublished diary that I had access to through the courtesy of the family. And so the Sanger turns up to Cambridge in 1936 at St John's College. It's one of the bigger and richer colleges in Cambridge. And he's introduced to biochemistry by meeting his tutor. He claims he didn't know anything about biochemistry. This is the man who introduced Fred Sanger to biochemistry. His name was Ernest Baldwin. He was at the time a lecturer in the Department of Biochemistry in Cambridge. And Sanger said to me, when I interviewed him in 1992, fancy being able to study biology using the methods of chemistry, which is as good a definition of any of the word biochemistry. I hope you would agree. So, but there were problems. Both Sanger's parents died when the young Sanger was an undergraduate at St John's College. And the other problem was here at the bottom. Sorry, I've got out. At the bottom here, all right, Sanger took 4 years to graduate at Cambridge instead of the usual 3, because he didn't do well in his first year in Cambridge, getting only a 3rd in the prelims. So here we have a man who's a double Nobel laureate who did very badly in his first year at university. Most people don't know this. And so don't be worried if your children, grandchildren don't immediately appear to be as academic as you might hope. And of course, with other problems, the Sanger parents had paid for him to go to university, so a guardian had to look after his inheritance because until Sanger was 21 years old. So this was a difficult time for Sanger, deaf his parents. And the reason he didn't do well in the first year, he took the wrong subjects. He took physics and maths, which he hadn't done so well at school, whereas he really liked chemistry and biology. So an important factor in the young Sanger's life was he met his future wife, here, Joan Sanger, an economics student at Newnham. And they met through an interest, a pacifist interest, because the Quakers believed in pacifism. They didn't want to fight. And Fred Sanger himself became what was called a conscientious objector, and he was excused from war service, because we're talking now about 1939-40, just before the beginning of the Second World War. So this is a picture of the young Sanger and his wife at about the time of their marriage. It's a lovely picture, isn't it, of a happy young couple. So then Fred did his PhD at the biochemistry department, but he didn't actually think he was good enough. How did this come arise? Well, actually, he left the university after 4 years undergraduate, and he didn't think he was good enough to do research. He had no plans to do a PhD. So again, think of that. A double Nobel laureate had no plans to do a PhD. He was, I think, an unconfident young man. But he did change his mind when he learnt he'd been awarded first class, quite indirectly, apparently through reading something in the Times. And he wrote back to the professor of biochemistry at Hopkins, got no reply, wrote again, got no reply. So he just turned up. And because he had a legacy from his parents' estate, probably still managed by his guardian, I don't know, but worth a considerable amount of money in today's term, he basically chose his PhD supervisor. And so he did come back to Cambridge. He did start his PhD. So his PhD thesis occupied this period of time, and it wasn't on insulin, it was on the amino acid lysine and its metabolism. Again, Sanger later on said in 1992 to me, and he also wrote a sort of autobiography article in, I forget when, a review, and he said, I wouldn't say it's a great thesis, but actually it is. I've read his thesis, of course, after he died. I was given a copy of it, and it is a very critically written thesis, and he did publish 4 papers on it. So again, he was being immodest, and it also he criticised Krebs, the famous biochemist. He couldn't repeat some of Krebs's papers. So I mean, he did have a good supervisor. The supervisor's name was Neuberger, who Neuberger was a young man then, and Sanger was his only student. So he had a very good introduction to science in this period. But we now come to the insulin period. And it's not well known that Sanger did not set out to sequence insulin. This was his first. He was offered a position by the incoming professor Chibnall from London in '43. Hopkins retired then, to just look at the end groups of this protein of considerable medical interest, because of course insulin was the lifesaver for people with diabetes through the work of Banton and Best. And Sanger had a very limited objective, just to look at the so-called end groups of insulin. Its amino acid composition was known, but of course its size was, it was known to be a smallish protein, but its size was actually overestimated at the time. So I'm sorry, I now have the first chemical slides, which shows the reagent that Sanger developed, which is, I've called the first Sanger reagent, because of course Sanger developed a reagent much later in DNA sequencing, the dideoxys. So this was this small fluorodinitrobenzene which reacted with, this is the end group of a protein, the protein going here, under quite mild conditions to form a derivative, a coloured derivative, which on the treatment with strong acid, 6-normal HCl, released this derivative, a dinitrophenyl amino acid, a coloured, yellow-coloured compound. And Sanger took advantage of the technology being developed at that time by Martin and Singh, the technology of paper chromatography, and especially two-dimensional paper chromatography, partition chromatography, to use the up-and-coming methods available then to identify dinitrophenylalanine and dinitrophenyl glycines as end groups. But so a critical observation he made at that time was, because of the instability of dinitrophenyl glycine to acid, it degraded to other compounds, and he didn't get equimolar yields of this compound. He had to modify the conditions, and he used partial, milder acid treatment. And so this was the start of the sort of partial acid hydrolysis procedure for looking at insulin. He basically found dinitrophenylalanine glycine attached to peptide, to 2 or 3 other amino acids. And so he was guided by his own observations, making rational deductions. And about 1949, I uncovered clear evidence that he himself had decided that he could sequence insulin. But even then, it was a very bold statement, because he had to separate the 2 chains. Here you have the final result. The the two chains of insulin join. And he had to sort out how to find out how these were linked through these disulfide bridges through cysteine residues. And he had the equally difficult problem because of working out where I'm using the old nomenclature here for amino acids. This is the published sequence in 55, when the three-letter code for amino acids was used, not the one-letter code that we now use. I had a difficult problem of deciding which amides, because the acid broke the bond and you couldn't distinguish glutamic acid from glutamine or aspartic acid from asparagine. So this is a real milestone. It's very difficult now to appreciate what a milestone this was, as Uti was saying, because this sequence had no obvious repeating elements. There was no way of predicting this sequence. And so the conclusion came that this was a unique sequence. There are no general principles defining the sequence of insulin or by implication any other proteins. And we still can't predict the sequence of a protein, as Uti was implying. We know the shape is important, but even then we can't go from we can't, or maybe I'm wrong, perhaps there are clever scientists around now who can go from a sequence and predict how it falls into a shape. So this is a very important principle at the time, that proteins had a unique sequence. I mean, I actually asked Sanger, did it have a unique sequence? Could some of these glutamines be glutamic acids? And he admitted they could have been, because he admitted his techniques to me, and this is recorded in the book. I'm sorry, there's the sales pitch of my book published in 2014. He admitted that that Sanger admitted his methods were not completely quantitative. Oh, I've gone. I beg your pardon. So we now go on to the next stage. Yes, so I'm introducing here what I call the Brownlee Brenner controversy. I mean, I shouldn't link my name to the famous Brenner, but this arose because Brenner wrote a review of my book for RNA, where by implication he criticised my statement that Sanger's knowledge, Sanger's observation that proteins had a unique sequence was not really important in the development of the ideas of the genetic code by Crick and others. And so I just want to share with you two comments which I think negate what Brenner said. I mean, it's unfair to Brenner. Brenner died recently, so he can't come back at me. But Ambler told me about informal talks that were held. Ambler was a student of Sanger. Told me about informal talks held in Francis Crick's house in the evenings in the 50s, where he, Fred Sanger, Sidney Brenner and Max Perutz attended. And their purpose was to allow discussion of how genetic problems that Francis and Sidney were interested in could be solved by Fred Sanger's methods of sequencing proteins. And the other thing that happened about this time that I think Brenner must have forgotten about when he reviewed my book was the very important work of Vernon Ingram in 1956. Using the Sanger micromethods of sequencing proteins, he studied the beta globin chain of a hemoglobin in patients with sickle cell anemia. Sickle cell anemia is a disease prevalent in Mediterranean areas and Far East, in which it's an inherited condition where the red blood cells have an abnormal shape, and patients with it, certainly before modern treatment, did not do well. But the point is, Vernon Ingram used Fred Sanger's fingerprinting methods to find a single amino acid change of a glutamic to a valine in the beta globin chain. This was the first ever known example of a single amino acid change causing disease. So I think the logical conclusion from Ingram's work is that DNA sequence must be translated to amino acid sequence. And we have to remember in 1955-56, as Uti said, there were conflicting. Nothing really was known about messenger RNA. Nothing was known about protein synthesis. In fact, some people thought protein synthesis was at that time was the reversal of proteolysis. So did Sanger have a philosophical approach to science? So the only thing I've managed to collect in my, I suppose, historical work on Sanger, I'm not a historian, I'm a molecular biologist, is Ambler told me Fred Sanger was not interested in philosophical issues. He did not, these are not my words, these are Ambler's words. He did not waste time pursuing unlikely hypotheses. He was not influenced by Popper and Medawar. And he went on to say, actually, I haven't put it here, he went on to say Fred Sanger's really interested in methods didn't engage him as a hypothesis, although I would contend that Sanger did have a hypothesis. His hypothesis was it was possible to sequence a protein in 1940 when no one else thought it was possible. And later on it was possible to sequence a nucleic acid when no one else thought it was possible to sequence a nucleic acid. So his hypothesis was, if you like, a methodological vision. So this is the young Sanger looking very happy as a young man of 40, awarded his first Nobel Prize. And he had no aspiration to change. He didn't want to influence scientific policy in Britain. He just wanted to go on and doing research. So what did he do? Now here he did set out to sequence RNA, the RNA period. DNA, I joined him in 1963, and actually I was unsuccessful in the first year in purifying phenylalanine tRNA by a clever method. Although I was successful, I was seconded to the laboratory of Robin Monroe, where I met my wife. So that was a success. But but then after 6 months in this lab, I said to Sanger, I'm not making any progress. I think I dropped several tubes. Fraction collectors were not very reliable in those days, or I wasn't very reliable as a young student. So Sanger had to concede to Holly the Nobel Prize for the first ever RNA sequence. I suppose had I been successful, and again Brenner brought this up in his review of my book, he said Brownlee was unsuccessful. We might have just have beaten Holly, although I think it's doubtful because Holly had a good start. But I did introduce the 5S ribosomal RNA as a model for Sanger and I to develop radioactive sequencing methods. This brings me to this RNA period. Sanger had a really good idea, which was to use radioactive sequencing. Holly had done all this sequencing by using the UV absorption of the 4 bases in RNA, cytosine, adenine, guanine, and uracil. A pretty insensitive way, where Sanger wanted to use the methods, the micromethods he was used to for working with proteins, and so his idea was to introduce a radioactive isotope, P32. And this was a successful idea, and it really was, as far as Sanger was concerned, I believe, although I never personally discussed this with him, but it was really a continuation from his protein work, particularly his idea of developing micromethods. And in those days we had to, you may not believe, we had to manipulate 1 microliter volumes. So I learned as a student to manipulate quite small volumes. I mean, I know it's now much easier, but most people even now will not know how to work with 1 microliter of solution or set up a reaction with 2 or 3 microliters. They'll be quite happy to set it up with 10 microliters or 0.1 of a mil. So we learnt that working in Sanger's lab. And so this is, I guess, my contribution, and Sanger's, because I needed help from Sanger and Bart Burrell to complete this sequence. So this is the sequence of the 5S ribosomal RNA, which was the first sequence done by these radioactive methods. But let me not dwell on that and come to a point which touches on Fred Sanger's ethics. Fred Sanger was very keen to acknowledge the help of his technicians. This is Bart Burrell on the left, who went on to work with on the Epstein-Barr virus DNA sequence, but early on was introduced to the Sanger way of thinking in his sequencing by Sanger. This is Alan Coulson, also who came to work with Sanger as a technician. Really no experience. These weren't PhD students, although Bart Burrell did do a PhD latterly. I can't remember whether Alan Coulson did, but they both became his long-term assistants. And Fred Sanger valued this contact. He said it's very important to work with someone you like. They don't, and who you get on well with. So this was also a very important feature. Sanger, he wasn't he wasn't a very expressive man. He didn't chat with you, but you felt you could get on with him, and he got on with. There are not many people he didn't get on with. And so he gave credit, as I said, to the technical support from Alan Coulson. So he was effectively mentoring them. And as I said, Bart Barail went on to do independent work on Epstein virus DNA when Bart Barail went to the Sanger Centre that was established much later. And Coulson, the man here, Coulson on the right here, he actually wrote more papers with Sanger, was an author on more papers with Sanger than any other scientist. Although Coulson didn't initiate research, but he was just a long-term colleague. So before I move on to the DNA work, and I hope I haven't overstepped my time, I just want to tell you one of the things that Sanger was very pleased to have found out. He did the first actual sequence of a messenger RNA. This is the coat protein sequence of part of an RNA phage. It's called the R17 bacteriophage. Phages, as you know, are the viruses that attack bacteria. And this was the first ever sequence showing, in fact, that the genetic code worked out about 65, this is 4 years later, was correct. So, because the genetic code in the, by Nierenberg and Khorana was worked out by very indirect methods, very clever methods. But this was the first direct sequencing of messenger RNA, and this was done by uniformly labeling the RNA phage. So the DNA period, arguably the most important work that Sanger did. When I started with Sanger in '62, I didn't know that Sanger had a vision to sequence DNA. I mean, DNA, there was actually no short DNA available, even then in '73. So DNA was in sight, but this was because Fred Sanger was continuing with different postdocs and students testing different ideas of how to sequence DNA. But the method that actually worked and produced the clear results was a copying method, where you copied DNA, essentially interrogating a long DNA, maybe many thousands, tens of thousands of nucleotides long, with a very short primer that formed Watson-Crick base pairs with a specific region. And you incorporated the radioactivity. Sanger was working still on the idea of radioactively labeled DNA. You incorporated DNA by having a phosphate labeled deoxynucleoside triphosphate. And so you incorporated the label as a copy. So this is the idea that which was very difficult to do then, because it was very difficult to synthesize very short oligonucleotide primers. Now, of course, you can dial them up and get them next day from a company. But it took Kossel, a colleague of Sanger, a year to make this short oligonucleotide primer to interrogate another bacteriophage DNA that Sanger had started to work on. It was the so-called Phi-X174 bacteriophage DNA that Sadat had introduced to the Sanger lab from the Sinsheimer lab in Caltech. Was Sinsheimer Caltech? Yes. Thank you, I got that right. So this is the critical slide of this Sanger method eventually developed. The idea was to use a copying method with a radioactive triphosphate. I should have labeled this phosphate with P32, but this is deoxynucleoside thymine triphosphate. And Sanger's idea was to work with a mixture of this triphosphate in that copying reaction I showed you down here. A mixture of this triphosphate with an analog which was known to be incorporated into DNA, and this was an analog that had been worked on by Kornberg and his colleagues who were interested in DNA synthesis, and it simply differed from the triphosphate in having a hydrogen atom replacing the hydroxyl group, and so it became a chain terminator. So if you had a mixture of these two compounds in appropriate proportions, this being radio labeled, this not, you could get and you used gel technology, which was the other thing Sanger introduced, the idea that a acrylamide gel could separate single nucleotide differences, and this shows a sequencing reaction carried on. I can't expect you to read it. 10 different reactions, not only with as I said, the mixture of deoxythymidine triphosphate and dideoxy1, but with the other 3 pairs, that is a deoxyguanine triphosphate and a dideoxyguanine triphosphate, deoxyadenine triphosphate, and the other one. And of course, this was difficult for Sanger because he had to go and synthesize these. Although this was available in the literature, Sanger had to synthesize, and Coulson had to synthesize, this side, the dideoxy derivatives of guanine, adenine, and cytosine. And this actually took Sanger a year to do. So this was a considerable hurdle to Sanger, and it's a credit to him. And I think his chemical knowledge that he had learnt as a young man, that he felt able to tackle this. He didn't. I mean, I think he did initially want a drug company to do it, and they sort of failed, and so he just set up to do it himself. What's happened, so I now come on to, so this is arguably, I think, the most important of Sanger's contributions, other than that first concept that proteins had a unique sequence. And as many of you will know, the Sanger method was, in fact, the dideoxy method was, in fact, the method that was used right through to about here, but all these sequences that emerged long after Sanger retired. Sanger retired in '83. So these genome sequences were done essentially using the Sanger method, of course modified to have fluorophores attached to the 5' position of the nucleoside triphosphates. And there were other, of course, automations done. Of course, since then, a lot has happened to sequence. I think we're in the 4th generation. Those of you who are doing DNA sequencing will correct me now. For these more modern sequences, this one is one of the latest to show 10 times the genome complexity of Homo sapiens. These use high throughput methods where you simultaneously sequence in a solid state DNA that is attached to a solid matrix. And the methods are too detailed for me to go into, and I probably don't understand them fully myself now. But so the methods have changed, although one of the most important methods by the company Illumina still uses dideoxys, albeit in a reversible form, as part of their methodology. I know now those of you who sequence, or if students sequence, they don't actually know what's the sequencing, how it's done, because it's outsourced to a company. But so the dideoxy principle is still part of the principle of the Illumina methodology. So this is my sort of summary. I hope I haven't gone over time. I probably have a few minutes. Sanger was really, I would call Sanger an experimentalist. He was really interested in developing new methods. He had vision. I suppose you could translate the word vision into philosophy. He knew that sequencing would open up whole new areas of research. He was a very pragmatic man who didn't easily give up, and he had many failed experiments. You can look at his notebooks, they're digitized at the Wellcome Trust Centre in London. And he was always in the lab right up the day before he retired. He didn't spend much time in an office. He actually didn't spend much time putting his sequences onto computers. He left others to do that. What about his personal traits, or what I call his ethics? I'm sorry, there'll be people here who criticize my use of the word ethics. I suppose I'm using it because of, probably imprecisely, but Sanger never over-directed others. I mean, he could never have been accused of bullying. This is a theme now in the department in which I work. I see notices, no bullying in this department. Our head of department, although I'm retired, I still go into the department, I'm surprised there's an anti-bullying campaign going on in Oxford University departments. I don't know if it's happening in Israel. I think the problem is that students come under too much pressure, PhD students come under too much pressure from their supervisors, but I don't know. I keep well out of it. But some students and even postdocs were surprised they got little direction from Sanger. And I think this was part of his mentoring style. He always wanted students or postdocs to take the initiative. And that was because he was training them to become leaders. And Sanger was a leader. There's no doubt about it. Now, I wrote at the end of my book, Sanger, I believe, was one of the greatest scientists of all time, whose name must be linked to Madame Curie and Charles Darwin because of the impact of the discoveries they make and will continue to be made. And I think there's no better legacy that Sanger has. Sanger died, these are three of his students who became famous. Rodney Porter, actually his first student in the protein field, who discovered the structure of antibodies. Cesar Milstein, who wasn't formally a student of Sanger, he was a student of Dixon, but got considerable influence from Sanger while Sanger was in the Department of Biochemistry, who I'm sure you know invented monoclonal antibodies. And this is Elizabeth Blackburn. All these who worked, an Australian student who worked with Sanger in Cambridge and was honoured for her work while at Yale, I believe it was, for work on telomeres and telomerase. So is there any better legacy? I think not. Thank you very much, and I'll be pleased to take questions.
Any comments, philosophical comments or historical comments or otherwise? Yes, Uta.
Thank you very much. First of all, George gave me this book as a present, and if anybody wants to have a look at it. The microphone. Okay, so here is this book, and who wants to look at it, I'm happy to loan it. Borrow, not to loan. Okay, I have a question. First of all, I think you described quite precisely this philosophy. And
I'm pleased that, tell me what it is.
I don't tell you later, I mean, I don't want to over. But my question is, why was he driven to sequence proteins? I'm not talking about DNA, but why was he driven to sequence proteins? You said did he want to contribute to open up new fields of research, or was there a thing that it might be important?
He was introduced to insulin by an incoming Professor Chibnall, and Sanger needed a job because Albert Neuberger, his supervisor, had moved to London. I didn't say this, actually in Sanger's third year as a PhD student in Cambridge, his supervisor had moved away, and Sanger didn't want to move away. He just got married to Joan, his wife, so he needed a job, and so Chibnall, as far as I know, and as far as Sanger's told me, Chibnall said, here's some insulin, look at its end groups. He wanted, Chibnall had not, he knew his team had worked out which amino acids were there. I think 16 of the 20 amino acids are in insulin. If I remember, there's no tryptophan, and I forget which other amino acids are missing from insulin. So, if you like, it was an accident that he was introduced to insulin, and the decision to sequence insulin came a bit later in 1949 when he observed, again I obviously went over this a bit quickly, when he observed that he could isolate a DNP glycine linked to the next amino acid, so he realized he could use this fluorodinitrobenzene method to along with compositional analysis. If you have essentially a dipeptide, you know the end group, you know the sequence of the dipeptide, and so he extended this. So he didn't set out to sequence insulin. He decided himself about 1949 that it was possible using his technology, the earliest Sangha reagent, fluorodinitrobenzene, and the methods introduced by Martin and Singh, and further methods that he introduced, paper electrophoresis. That was then he decided to sequence. So if you like, he was fortunate to have been given a good problem by Chibnall.
Two comments and then a question. The first comment is concerned with both you and Singer finding your spouses in the laboratory. I mean, perhaps people get the wrong idea. They shouldn't send their children to do the art, they should send them to the sciences if they want to get married. That's one question. That's one
comment, is it?
Second comment, and this I owe to a friend of mine who passed away recently, who actually read your book and came over rushing to me and said, look, this book shows something that people don't realise. And he believes that the message of your book, which you didn't mention, and that he took to be the message, is he accounts for the only man ever to get 2 Nobel prizes, although Curie was as well, but the only male, was he didn't specialise till later on. That is, he had many. His knowledge was of many different fields, and perhaps, and this is Joan said that the trouble with education is, and especially scientific, that people are made to specialise too early. The wider it gives you a choice, and what he tried to say in something that he wrote on your book, I don't know if it was ever published, but it was that it was because of his knowledge that he could take one idea from here and put it in a different area from here, and one methodology from here and put to that.
Do you want me to comment on?
Perhaps you'd like to just comment.
So is that the question? Did he have a very broad education? Was he capable of moving fields?
What students get today.
Yes. Actually, I think I'd have to slightly take issue with what you said. I mean, Sanger had a pretty good scientific background. He was I suppose you could argue, and I think Sanger told me himself, you could argue how was it he felt confident to move from the field of amino acid metabolism to sequencing of proteins, to sequencing of nucleic acids. So yes, people did comment, how could a man, why didn't he continue to work on proteins? Why did he change from proteins to nucleic acids? I've tried to emphasize the similarity, but equally, as you said, you could say, well, this is a man who has the confidence to move fields. So he moved fields, but he kept a sort of methodology, this micro methodology. What I didn't mention is he actually had the idea after sequencing insulin to use radioactive methods for sequencing proteins. Sanger called this period, 1955 to 62, his lean years, when he didn't publish much. So there is a continuity on the methodological front, but I do agree with you. He was a man who was prepared to move fields, and I think I learnt that from him. And I think last night I introduced myself as a lateral thinker. People say to me, well, how have I got the confidence to write a historical biography when I've never done that before? So I agree in part with what you said.